Okay, so we're in your notes in the carbohydrate packet, and go to the end of the fat section and you will see proteins there. Proteins are another one of the macromolecules that we've been talking about in organic chemistry. We said that it is a polymer, and its monomer or building block is the amino acid. It makes up about 50% of the body by weight, and you can see that there are a lot of different like funky shapes, and they're going to have a lot of weird functions. Today's topics we're going to cover is the amino acid structure, how these amino acids link together to make that polymer or that protein, how these proteins fold and what causes them to unfold, the relationship between a protein structure and its function, and then finally we're actually going to do the functions of proteins. So we're going to skip that section in your notes right now, go down to the part where it says building block. That's where we're going to begin. Okay, the building block is the amino acid, and there are 20 different types of these. The structure of them is about the same, except for this R group, which we call the variable group or the side group. This is what determines what the amino acid is. So there's 20 different amino acids, 20 different side groups or R groups there. There's a couple other functional groups in this that we've already talked about. We talked about the amine group, which we said was very basic, that it can accept hydrogen. In the center here, this is what we call an alpha carbon. And this alpha carbon is just in the middle, connecting the amine group, the hydrogen, the R group, and then of course the carboxyl group. Okay, we talked about that carboxyl group being acidic because it can donate its H+, which makes amino acids, aka proteins, great buffers. Different side groups, like I said, make the different amino acids, and so here's a couple examples of those. This is glycine right here. This is the simplest of the amino acids, and its R group is just NH, a hydrogen. At the bottom here, this is cysteine, and you'll see at the very bottom you have that sulfhydryl group or that thiol group. This is going to come up later when we talk about protein folding, because this is one of the ones that is involved in disulfide bridges. This one actually has a benzene ring in that. Okay, so it's kind of a complicated R group. And then the last one here, you can see at the very ends we have 2 methyl groups, which is going to make this amino acid very nonpolar. Okay, so these R groups can either be polar, they can be nonpolar, okay, or hydrophobic. They can be acidic, or they can be basic. And so these different characteristics are going to kind of determine how the protein folds. All right, let's look how the amino acids actually link together. We've talked about this a little bit when we talked about dehydration synthesis. Okay, remember that in one amino acid, the OH group is being donated from the carboxyl, and in the other amino acid, we have the hydrogen from the amine group. Okay, this is going to come together and form water. Okay, this is again a condensation reaction or dehydration synthesis. In your notes, I want you to write down that a peptide bond is a covalent bond. It forms between the carboxyl group of one amino acid and the amine group of another amino acid. You can see right here that nice covalent bond is going to form between the carbon and the nitrogen. This is going to be a very strong bond. And when we bond 2 amino acids together, we're going to call that a dipeptide. Okay, di meaning 2, peptide being the name of the bond. A polypeptide, which is a term you should probably write in the margin of your notes, this is when 3 or more amino acids are bonded together. Okay, again, poly meaning many, peptide being the bond. Okay, that is a long chain of amino acids. The sequence of amino acids determines the structure of the polypeptide, okay, and eventually the protein itself. Structure, in turn, determines function. Okay, so that's the next things you need to write in your notes. Sequence of amino acids determines the structure, and structure determines the function. Okay, as said on the previous slide, a long chain, or 3 or more amino acids, is a polypeptide. If we look at the sequence here, this is what we call the primary structure, or just a chain of amino acids. Top picture is kind of cartoon. The bottom one is showing all those peptide bonds. And at each end of the protein, we're going to have either a free amine group, which we'll call the N-terminus. Okay, so the N-terminus is the end of the protein where we find the amine group. Okay, that can bond to something else. On the other end, then, we have a free carboxyl group. And this is what we call the C-terminus, okay, or the end that has the carboxyl group on it. In your notes, the next blank says you should put polypeptide. A polypeptide is not equal to a protein. Okay, sometimes a polypeptide is equal to a protein if the protein is only made of one set of amino acids. Okay, and we'll come back and talk about this on another slide. Usually, though, a protein is made up of one or more polypeptides bonded together. Okay, proteins do have a three-dimensional shape. In fact, 2 shapes are probably the most common. One of them is fibrous proteins. Okay, fibrous proteins are long strands, and these are usually used for structure in the body. There's a protein in our nails called keratin. Okay, it kind of gives our nails that hardness and that waterproofing. It's also in our skin, and that is a great example of a structural fibrous protein. The second type is the globular protein, and these are a lot more common. These are going to have a variety of jobs. Okay, we're going to find a lot of them in the cell membrane. A lot of these are going to be enzymes. Okay, and they kind of have like the goofy shapes. All right, let's look at the 4 levels of protein structure and our folding. Okay, again, let's look at the picture before we take our notes. Primary structure there, that's going to be the first level. Okay, secondary structure, this is going to be the second level. Tertiary, the third. And quaternary, the 4th level. Okay, we'll start in our notes with the primary structure. Okay, primary structure is just the sequence of amino acids. Okay, it's what we saw in the picture before, and this is caused by those peptide bonds. Okay, those strong covalent bonds between the hydrogen and nitrogen of the 2 functional groups. The second level of protein folding or structure is the secondary level. This is caused by hydrogen bonds, and if you remember from before, hydrogen bonds are very weak bonds. However, if you have them in a large number, they can maintain a pretty good structure. These hydrogen bonds are actually going to make 2 shapes at the secondary level. The first one is what we call an alpha helix, and this is just a clockwise coil or like a corkscrew, and this is caused by hydrogen bonds between every 4 amino acids. The second type is what we call a beta pleated sheet. And a beta pleated sheet is when the protein folds back and forth, okay, just like forward and backward and forward and backward. And hydrogen bonds are going to form between the 2 parallel parts. So you can see in the picture here that the hydrogen bond is forming between 2 different strands of the protein. The third level of protein folding is the tertiary structure. And this is what causes the overall 3D shape of the protein. This is mainly caused by all of the interactions of the R groups, or the variable groups, on the different amino acids. Okay, so depending on the order they were, we're going to have the proteins folding in a lot of different ways. Okay, lots of types of bonds are going to be in this tertiary structure. Pretty much every type of bond that we've talked about. Okay, the first one being hydrophobic interactions. Okay, if you look at the picture here, here's one of those amino acids that have the methyl groups. Okay, if this is in our body and in water, that wants to be as far away from water as possible because it is so nonpolar. And so what's going to happen is all of these nonpolar R groups are going to fold inward and be at the core, or the center, of the protein. Okay, so in your notes, by hydrophobic interactions, amino acids with nonpolar R groups end up clustered at the core. Okay, they fold inward. Second type of bonds in your notes is van der Waals interactions. And we really didn't talk about these, but these are intermolecular bonds. Okay, so they are between molecules that are very, very close together. Okay, they're usually very weak interactions, and we're not even really going to talk about those. The next one is ionic bonds, obviously between a metal and a nonmetal. And if you look at the picture here, you'll see that this is actually between an acid and a base. Okay, you have your carboxyl group there and your amine group. So those acidic and basic amino acids are going to make up the ionic bonds. Next type is a hydrogen bond. Okay, again, a weak interaction. And we're going to see that between the hydrogen of one side group and another polar molecule of another side group. Usually it's going to be oxygen. And the last type and the strongest type is disulfide bridges. Disulfide bridges are covalent bonds. These form where two cysteine amino acids are close together. So we talked about in our notes those sulfhydryl groups. So here's the case where they lose their hydrogens and those two sulfurs come together to make this bridge. Okay, so this is a very, very strong bond. This is the strongest bond that's in the protein. The last level of protein folding is going to be the quaternary level. And actually, not all proteins are going to have this level. If a protein is only made of one polypeptide, it will stop at the tertiary level. Okay, the quaternary level is only when you have two or more polypeptides together. Okay, each one of these polypeptides is what we're going to call a subunit. And if you look at the picture to the right, this is a hemoglobin molecule, and it is made up of 4 polypeptides. So those 4 polypeptides come together to make this one protein. Okay, the bonds holding it together are going to be the same bonds in tertiary. So we're going to have some hydrogen, we're going to have some ionic, we're going to have some hydrophobic interactions going on, we're going to have some disulfide bridges. Okay, and this is going to be the final shape of the protein. Okay, so hemoglobin is our example, 4 polypeptides coming together, making one protein. All right, so this is a good place to kind of go back and talk about the difference between a polypeptide and a protein. Polypeptide is just a long chain of amino acids. A protein can just be a polypeptide, but this is when it's wound up and it has at least 3 levels of structure. Okay, so if you kind of think of a polypeptide like yarn, a protein is when you actually knit something from it. Okay, it becomes a sweater or a mitten or something else. Okay, usually that protein is going to be more than one polypeptide together. All right, in your notes, we talked about hemoglobin. Right now you just need to know that that is a protein that carries oxygen in the blood. Okay, it's a good example of quaternary because it has the 4 different polypeptides. All right, underneath that is a word called denaturation. Okay, this is when proteins unfold and lose their function. You'll often hear this used as a verb. We'll say that a protein denatures. It's the same thing. What it means is it starts to unwind. Okay, we're going to lose our tertiary and secondary folding levels. Okay, primary, or the sequence of amino acids, is still there, but the hydrogen bonds, the ionic bonds, those things are completely interrupted. There are several things that can cause denaturation. Okay, you guys have seen this when you make an egg. Now it turns from clear into white. That is the protein denaturing. It's unwinding. So the first thing that's going to cause denaturing is heat. Okay? Heat breaks up those hydrogen bonds really easily, and so we start to lose the protein shape. This is also why a high fever is really bad for humans, because over 105 degrees, all of our proteins start to unwind. Second thing that causes denaturation is a change in pH. Because we do have those side groups or R groups that are acidic or basic, a change into an acid or base is going to interrupt those ionic bonds. Those interrupted ionic bonds are then going to cause the protein to unfold. Another thing that could change this is if we change the polarity of a solvent. Okay, normally we think of our solvent as water, and so all of the non-polar things go to the center. However, if you change the solvent to be something that is non-polar, everything that is polar is going to go to the center. Okay, so that protein is actually going to flip inside out. And we'll see this in some cases for our proteins in the cell membrane. The last thing that can really cause proteins to denature is salts. Again, salts form strong ions, and so that's going to interrupt that ionic bonding and cause the attractions to be off. So any of those things can cause proteins to denature. All right, the next word in your notes is chaperonins or chaperone proteins. These are protein structures, and you can see they kind of have a hollow center, and their job is to help proteins to fold. So that polypeptide will actually go into the center where the arrow is pointing. Okay, we're kind of looking at the end view there, whereas the other one is a side view. And that polypeptide will go in there, and it kind of creates a good environment for the amino acid side groups to form all of their bonds. Okay, it's kind of a more protected area so that it can fold up well before it is released into the cell or into the blood. Okay, so these are protein structures that help fold other proteins. All right, finally, we're down to functions of proteins. So you actually need to flip back to the very beginning of your notes. And I want you to write the word down, and I want you to write at least one example of how these functions are used in a protein. All right, so the first function of proteins is structure. Okay, we already talked about keratin fibers in our nails, those long fibers, but also collagen fibers is a good example. This is what makes up our tendons and our ligaments, things that hold our bones together, things that hold our muscle to the bone. And you can see those white areas in the picture, those are actually the collagen fibers, okay? Strong structural proteins. The other part of the picture is muscle fiber, okay? Again, lots of protein making up that muscle. Second major function is enzymes. Enzymes are the catalysts in living things. Okay, the word catalyst is underneath the list of functions. A catalyst is something that speeds up a chemical reaction without being consumed. Okay, so a catalyst is really important for our metabolism. We actually wouldn't be able to survive without our enzymes because it would take so long for the chemical reactions to occur that we would die before things happened that we needed them. Almost all of the enzymes in the body are proteins. Okay, there are a couple that are made of RNA. We actually call them ribozymes, but for the most part, 99% are going to be enzymes. We'll talk about them more in the future. The third function of proteins is transport. Got a couple examples for this. First of all, we have proteins in the cell membrane. Okay, the cell membrane is very nonpolar, so in order to get big substances or ions or polar molecules in, a lot of times they're going to have to go through a protein. So here's kind of like a little picture of a carrier protein that moves an ion across the membrane. Another example is the hemoglobin that we looked at before, and that is one that carries oxygen in the blood. Okay, there are 4 irons attached to that, and so iron makes a ionic bond to the oxygen and carries it until it gets to a place where oxygen is needed. The fourth function of a protein is receptors. Receptor proteins, their job is to send and receive signals. Okay, so they're kind of like a communicator in our body. We have these in the cell membrane of every cell. You can see there's a little binding site. Usually a hormone will bind to that site and it will cause another protein to move. Okay, so this is all about communication. Okay, so again, proteins in the cell membrane. This is going to give our cells messages from their neighbors, from the brain, and things like that, so that our body can maintain that homeostasis. Okay, next to it, you also see a transport protein that kind of forms a channel. All right, the fifth function is motor proteins. Okay, these are ones that are going to cause movement, sometimes also called contractile proteins. Good example of this is going to be in cilia and flagella. You can see those little purple die-in arms. Okay, they actually contract and move down the protein back, and this is what causes the sperm to be able to swim. The same thing or a similar thing is also in cilia, okay, which are the little hairs that are outside a cell that can move things over the surface of our cells. We also have motor proteins in muscles. Okay, they contract, they move. And we also have things in our cell that's going to move our chromosomes or move the organelles around. Okay, those would all be great examples of motor proteins. The sixth function of proteins is defense against disease. And the example of this is an antibody. So here's a picture of a bacterial cell, and you can see on the surface there's all these different shapes. Those are proteins on the bacteria. And your body will recognize those as not being your cell. And so what it will do is it will make this Y-shaped protein called an antibody. Now, antibodies don't actually attack or kill the bacteria. What they do is they attach to it and they kind of tag it for death. They make it really big, and then they call the white blood cell, and the white blood cell will come and take that sucker out by eating it. All right, the seventh function is going to be hormones. A lot of our hormones are fats, but some of our hormones are proteins. Okay, a great example of this is insulin. We talked about the pancreas and how insulin regulates glucose and causes it to be stored in the liver or muscles as glycogen. Okay, that is a protein hormone. Also, growth hormone. This comes from our pituitary gland. This is what causes our bones to grow. This is what causes our muscle and cartilage to grow. It is a protein hormone. All right, and the final function for proteins is storage proteins. Now, we don't have a lot of storage proteins in the human body, but two great examples of these are going to be in plant seeds. Okay, this is a pea that's germinating. There are a lot of proteins in here that are stored that serve as food for that seed. Okay, there's sugars as well, but you're also going to have proteins. And then the last one is back to the egg white. That egg white, or albumin, is actually all the stored food for the cell that is growing inside the yolk. Okay, the yolk is going to be a lot of food as well. Okay, pretty high in fat, but the albumin is going to be a lot of protein that growing organism can use. All right, so that pretty much wraps up what we need to know about protein. Okay, so we talked about the amino acid structure. Most important thing is that R group that determines which of the 20 it is, and then our amine group and our carboxyl group that we've talked about before. Okay, we talked about how we link them together in a condensation reaction, how carboxyl gives up OH and amine gives up H to make a water, and then we make a nice covalent peptide bond between the carbon and the nitrogen. Okay, we talked about how proteins fold. We said that the sequence of amino acids really determines how it's going to fold, and we talked about the 4 levels that it can fold to. We also talked about denaturation, okay, or things that cause it to unfold, and we said heat, extreme acid, base, change of solvent, and salts are all things that can really affect our proteins. Relationship between protein structure and function we didn't talk too much about, but I'll give you a little clue. When we talk about this, if you change the structure of the protein or the shape, we are totally going to change the function. Okay, so if any of the amino acids get changed in the sequence, a lot of times the protein is not going to be able to work. Okay, we'll talk about this when we get into mutations in just a bit. And then finally, we covered the 8 functions of proteins. Okay, so just kind of go through those, make sure you have a good example and you can list those. All right, hopefully this wasn't too bad. I know it was a little bit long, but this will give us some class time to be able to do some activities.